Annual of Photography, 1942. The Visual Mechanism and Pictorial Photography by Lloyd E. Varden. The analogies between the human eye and photography are manifold and commonplace. As a matter of fact, the convenience in presenting the fundamentals of the photographic process by use of analogies with the eye has led to such a widespread adoption of the practice that almost every beginner in photography can describe the similarity between the camera lens and the lens of the eye, the iris and the diaphragm, the film and the retina, depth of field and visual accommodation, and so on. Photography and the human eye have not been associated in this manner alone. It is generally assumed that the ideal in photography is the reproduction of a scene in keeping with the way the eye sees it. For example, the response of a photographic emulsion to colors is popularly evaluated in terms of the wavelength sensitivity of the human eye, and the illusion of depth by proper perspective relations is said to comply with the visual image because of the true drawing effected by modern camera lenses. Briefly, among most practicing photographers, a relatively simple relation has been considered to exist between photographic recording and visual perception, and a physical correspondence between visual and optical imagery has been constructed. It is natural to expect that correlations of the above sort should be made. Man in general possesses the inherent characteristic of wanting to establish standards of perfection. His efforts must be directed toward goals which are definable within the realm of experience. Such goals are not limits but merely stepping stones. In setting up the standard for photographic perfection in terms of visual perception, one was chosen that is admirable, but more complex than would appear upon the surface. Visual perception is far more than a mere transfer of an optically formed pattern from the retina to the brain. This is a fact long known. However, for one reason or another, the complexity of the visual process has been alluded to only in the most scientific of photographic writings, and not in all of them. As a result, practical photographers cling to the belief that photography is a process which should, at its best, create images that reestablish a perfect correspondence with the visual impression. It is not unlikely that photographers have assumed a criterion of perfection that in many aspects is undesirable, if not unattainable. The criterion is undesirable in one respect because it has it. The criterion is undesirable in one respect because it was adopted without a complete knowledge of vision, which in turn led to a naive outlook in many instances, and in addition perpetuated unfounded arguments, especially arguments directed against other fields of art. By way of example, there is the age-old debate between photographers and painters concerning the correctness of drawing. The artist has claimed that because of his feeling and experience, he is able to relate the sizes of objects and their placement and shading to effect a better perspective than is possible by mechanical means. The photographer belittles this argument, contending that the lens of the eye and that of the camera produce the same type of image, and that since both images are formed by optical refraction, the camera image must be a more faithful rendering of the subject matter than an artist could ever hope to produce. Such assumptions are not at all supported by fact. Unquestionably, a highly corrected photographic lens will produce a more perfect image than an artist can approach, from a standpoint of detail and technical accuracy of drawing. But the supposed criterion is that the photographic image is a duplicate of the visual image. It is mostly because of the limitations of the visual mechanism that the artist is unable to match the detail perfection of the photographic image. These limitations are reflected in his result and perhaps lead to a better likeness of the visual impression. Furthermore, the mental perception of a scene does not necessarily, perhaps never, correspond with the dimensional proportions of objects as projected upon the retina by the optics of the eye. For example, a 6-foot man 20 feet from an observer will form an image about 0.3 inches high on the observer's retina, assuming the focal length of the eye to be 1 inch. A camera with a 1-inch lens will also form an image 0.31. A camera with a 1-inch lens will also form an image 0.3 inches high. Another 6-foot man at 100 feet distance will be imaged by the eye and the camera only 0.06 inches in height. This shows a perfect correlation between the two optically formed images. As pointed out by Thoulis, however, what the observer sees, that is, his perception, will be two men not nearly so different in size as indicated by the linear differences of the retinal images. Accordingly, lest photographers continue to place too much emphasis upon an ill-established criterion, it seems desirable and timely to examine the nature of the visual mechanism. A knowledge of some of the characteristics of the eye, both structurally and functionally, will do much to lessen the dogma that has been built up around the photographic visual correspondence. It will also make possible appreciation of certain fundamental limitations in this supposed idealistic correspondence. The structure of the human eye, evolutionary development. The human eye is one of the most complex structures of the body. It has developed through a long series of evolutionary processes, and like other complex body organs, it is only through a tracing of the major levels of development that one is able to perceive how such a specialized mechanism could evolve. To begin with, all protoplasmic substance is light sensitive to some extent. In the lower unicellular organisms, the response to light can be observed even though an organ of vision is absent. That's pretty cool. I'll say it again. In the lower unicellular organisms, the response to light can be observed even though an organ of vision is absent. In the multicellular animals, groups of cells become specialized in their response to light. For example, in the lowly Aurelia, common jellyfish, there are localized pigmented surface areas packed with nerve cells. The pigment acts as an efficient light absorber and is an accessory organ to the nervous tissue. In the higher evolution of the eye, one finds that the sensitive areas and accessory organs are equally important, both very specialized and structurally rather independent, but functionally cooperated to the highest degree. Various steps in the development of the eye are shown in Figure 1. These examples have been compiled from various references, but any number of similar sets could be chosen to show the same evolutionary development. Interesting to note is a marked parallel between the development of the human eye and the eye of the higher mollusks, the cephalopoda. In the eye of the nautilus, for example, a fluid is found in the eye cavity. The next stage is the formation of the lens, and finally, in the octopus and cuttlefish, the cephalopod eye finds its highest level, not far removed from the human eye. The human eye. In Figure 2 is a detailed drawing of the human eye. Each part will be discussed separately, but completeness of description will depend upon functional importance from a photographic standpoint. A great part of the eyeball is covered by 3 successive layers: the sclera, the choroid, and the retina. The sclera is a tough, thick, protective outer layer. The choroid is a center. A great part of the eyeball is covered by 3 successive layers: the sclera, the choroid, and the retina. The sclera is a tough, thick, protective. The sclera is a tough, thick, protective outer layer. The choroid is a center vascular membrane, thin and dark, to function somewhat as the non-halation backcoating on a photographic film. The retina is the inner layer covering the entire posterior chamber of the eye, consisting of a complex nerve structure along with special sensitive elements. At the anterior boundaries of the eyeball, the sclera merges with the equally tough but clear cornea. The cornea is so transparent it is hard to see, but upon careful examination it is observed to be a convex layer, which is the first refracting surface in the optic system of the eye. The choroid layer not only acts as a light absorber to prevent scatter, but it also provides nutritional elements to the eye through its highly developed vascular system, through which blood circulates. The choroid thickens near the front of the eye and, in ring fashion, extends completely around the cornea. This ring is the ciliary structure, and because of its radical muscular makeup, performs the very sensitive and specialized task of accommodation. The ciliary body cannot be seen when looking into the front of the eye because of a further continuation of the choroid, which passes over in front of the lens of the eye. This extension of the choroid is known as the iris and is the colored ring as seen from the front of the eye. It is equipped with independent muscles to automatically produce contraction and dilation through a range from 2 to 6 millimeters when the external light intensity is varied. Although the inner layer of the eyeball, the retina, is next in order for discussion, its description is best left until a few comments are made in regard to the optical system of the eye. Behind the cornea and iris is a chamber filled with a transparent fluid called the aqueous humor. Separating the aqueous humor from the inner chamber of the eyeball is the crystalline lens, double convex. The crystalline lens is located immediately behind the iris and is supported in a capsule to which are attached connective ligaments going to the ciliary body. It is this mechanism that controls the shape of the elastic lens. Behind the crystalline lens is located the inner chamber filled with a thick, non-liquid vitreous humor. The cornea, aqueous, and vitreous humors and the crystalline lens all contribute to the cornea, aqueous, and vitreous humors and the crystalline lens all contribute to the dioptric system of the eye. Their respective descriptions, as presented here, should not lead one to believe that they are homogeneous structures, for it is known that the crystalline lens consists of laminae, like layers on an onion, and is fibrous. There is also reason to believe that the vitreous humor is supported by a crisscross mesh. These facts add to our understanding of certain diffraction characteristics of the eye. The axial length of the human eye from the anterior corneal surface to the retina varies from around 20 millimeters to 36 millimeters in different individuals. In a normal eye, the average length is about 24 millimeters. The radius of curvature of the anterior surface of the cornea is from 7 to 8 millimeters, while the posterior surface is usually less than 7 millimeters. The thickness of the cornea is about 0.5 millimeters, and its refractive index is approximately 1.376. The axial thickness of the aqueous humor is roughly about 3 millimeters, and the index of refraction is 1.336. The thickness of the crystalline lens is about 3.6 millimeters and remains constant even during accommodation. The anterior radius of curvature of a relaxed eye is about 10 millimeters, and that of the posterior surface 6 millimeters. Actually, it is impossible to assign a single index of refraction to the crystalline lens, since each layer has a different value. At the vertex is found the lowest value, around 1.388. At the center, a maximum value of about 1.411. Sometimes a total index is assigned, which is the index of an optically equivalent homogeneous lens. This value is calculated to be 1.42. The total refracting power of the human eye, when totally relaxed, is of the order of 59 diopters. The diopter of a lens is the reciprocal of its focal length expressed in meters. This is often called the power or strength of the lens. The retina. The retina envelops almost the entire inner wall of the posterior chamber of the eye. It is in this part of the eye that light is registered, setting up nerve excitations, which are transferred to the sensorial regions of the brain, where the stimulus is interpreted. The retina is a thin membrane of about 0.2 millimeters thickness, which light can traverse with ease. The microscope reveals that the thinness of the retina is no indication of its complexity, for at around 1500 magnification, a transverse section may show up to 10 distinct layers. The function of the various layers is not too well understood, although each has been the subject of thorough study. Fortunately, a complete knowledge of all the retinal layers is not required for this discussion, but a few are of extreme importance. In Figure 3 is shown a simplified schematic drawing of the retinal layer. It can be seen that, contrary to one's expectations, the light focused upon the retina must pass through the entire layer before reaching the elements of sensitivity. This fact seems to be no barrier to good vision, however. In the foremost posterior region of the retinal layer are situated the so-called rods and cones, both of which have been considered the receptors of the visual organ, although at least one outstanding authority claims that only the cones are recipient bodies. The rods are shaped like thin cylinders, while the cones are bulged at one end and taper off at the other. These sensitive elements are highly concentrated but are not equally distributed over the entire retina. in the general region of sharpest vision called the macula lutea, or yellow spot, wherein is located the fovea centralis. The cones widely outnumber the rods. The number of cones in the macula has been established. The number of cones in the macula has been estimated as approximately 150,000 per square millimeter. There are actually no rods to speak of in the macula, and so it is easy to calculate the probable diameter of the cones in this region as around 2.6 micrometers. As counts are made farther and farther away from the foveal region, the number of cones per square millimeter decreases, the number of rods increases, and the total number of sensitive elements per unit area decreases. At the extreme periphery, the cones again become predominant. The cones also become larger outside the fovea. The total number of cones in the retina is of the order of 3 million, while according to some estimates, there are up to 20 times this many rods. It will also be noted that this retina is depressed in the foveal region, but it is questionable whether the thinness of this portion of the retina can in any way account for sharp vision. Some hawks have much better vision than man in spite of considerably thicker retinas. Since the retina covers the entire inner posterior wall of the eyeball, the angular scope of human vision for both eyes is almost 180 degrees in the horizontal plane. One can test this by stretching his arm out sideways and observing the wiggling of a finger while looking straight ahead. The vision at such an acute angle is not distinct, but it does exist. Actually, the angle of sharp vision is very small, since high visual acuity is limited to the tiny cone-packed oval-shaped fovea, having the approximate dimensions of 0.24 millimeters by 0.3 millimeters. The angle subtended in sharp vision is about 1 degree. Beyond this, vision is indistinct, and for the eye to cover an area subtending an angle greater than 1 degree, giving the impression of sharpness, it must scan the area continuously, such as happens when viewing a motion picture screen in a theater. We are not conscious of the scanning operation and are led to believe that we have a wider field of sharp vision than is actual. The excitations created photochemically in the receptors are conveyed through the retinal structure by a complex system of ganglion cells and nerve fibers. The cones of the minute fovea lead out to the optic nerve bundle by single fibers. However, since the total number of nerve fibers in the optic nerve leading to the brain is only about 100,000, it is obvious that each of the several million light-sensitive elements does not have a one-to-one correspondence with the brain. Actually, large groups of rods and cones are connected to a single nerve fiber, and this is further complicated by a most intricate visual path through the retina. Large groups of rods and cones are connected to a single nerve fiber, and this is further complicated by a most intricate visual path through the retinal layers. This can perhaps help to explain why retinal stimulation at one point often gives rise to adjacent disturbances. Light action on the retina. Although the more classical and more widely acknowledged theory, the duplicity theory, states that both the cones and the rods respond to light stimuli, they are said to react quite differently in most functional aspects. To begin with, according to this theory, the rods are very much more sensitive to light than the cones, and at very low levels of illumination, only the rods will respond. These levels are lower than the so-called cone threshold, which is around 0.001 lumens per square foot. The absolute threshold below which even the rods are non-responsive is about 0.00001 lumens per square foot. At low illumination levels where rod vision is predominant, it is generally assumed that no hues can be seen. Thus the expression, all cats are gray at night, this is known as night vision or scotopic vision. However, in spite of the fact that this assumption is well rooted in the field of psychological optics, the reader is warned not to accept it by any means as final. Some evidence points to the fact that the rods are not recipient bodies at all, only the cones functioning in this capacity. The cones, according to the duplicity theory, have a special function in creating sensations of hue in daylight, or during photopic vision. As we have noted, the cones are tightly packed, especially in the foveal region, and so they are also responsible for our distinct vision. partly explaining why visual activity is at a maximum under daylight illumination levels rather than at twilight. During night vision, when the cones are inoperative, the point of sharp focus on the retina automatically shifts from the fovea to a region where rods are present. The exact mechanism of visual excitation is not completely understood. There's even the possibility that what is thought to be understood is incorrect. However, it is known that there are light-sensitive substances present in the retina, which decompose photochemically, the ionic products of the decomposition acting indirectly as nerve activators, which create coordinated impulses that are sent to the brain via the optic nerve. One of the light-sensitive substances of the retina was discovered as far back as 1876 by Boll. This substance is known as visual purple, or rhodopsin. It is a complicated protein, purplish in color, quickly bleaching when exposed to light. The visual purple is found in the rods and in other parts of the retinal layer, but not in the cones. Experimental evidence of a corresponding light-sensitive material in the cones is scarce, but comparatively recently has been disclosed that a substance called iodopsin is present in them and may be responsible for their stimulation. Dr. Wald of Harvard has also isolated and identified 3 color pigments in the retinal cones, which no doubt are in some way connected with the photochemical cycle of vision. The pigments isolated by Dr. Wald are astatine, coloring pigment of a boiled lobster, xanthophyll, coloring matter in egg yolk, and carotene, the pigment of the carrot. These compounds are related to the vitamin A structure, which we shall see is of fundamental importance in the photochemical retinal process. The bleaching of the visual purple is directly proportional to the intensity of the light stimulus and to its concentration. If we call the light-sensitive substance S under the influence of light 2 decomposition product, If we call the light-sensitive substance S under the influence of light, 2 decomposition products, A and B, will be formed, thus S light, A plus B minus. Since in the presence of light the supply of S is rapidly diminished, there must be a reverse reaction taking place to restore the supply. It has been found that the ionic components A and B are recombined in the bloodstream, provided there is an ample concentration of vitamin A. Actually, one of the decomposition products of S is thought to be vitamin A, and the other a substance known as retinine. When there is a surplus of vitamin A, it is natural to expect the above reaction to be reversible. A plus B minus S. However, a more satisfactory explanation of the cycle has been given by Wald and Clark. They have viewed the decomposition and reformation of rhodopsin as taking place in 2 phases, one of which is reversible. The primary action of light upon the rhodopsin is the formation of retinine, visual yellow, and a protein. The retinine then forms vitamin A, which reacts with protein substances to reestablish rhodopsin, or the retinine reforms rhodopsin directly. This is shown in the equations below. Rhodopsin, light, retinine and protein, vitamin A and protein, creates rhodopsin, creates light, creates retinine and protein, creates vitamin A and protein. The sensitivity of the eye is dependent upon the concentration of S. The higher it is, the more effective a weak stimulus will be. However, the actual stimulation of a nerve fiber is not thought to be caused by one of the byproducts of the breakdown of S. Resting in the receptors, according to Hecht, is a substance which we shall designate as R. The R substance has a tendency to change to a nerve irritant, which can be represented by the letter I. But the reaction R to I takes place only in the presence of A plus B minus, and will not take place at all until the concentration of A and B has reached a definite value. There are certain justifications for Hecht's postulation. It explains why there is a lapse of time between light stimulation and sensation. It also contributes to the explanation of retention of image, so important for the production of motion pictures. It also explains why the eye is a poor criterion for judging duration of light flashes and should discourage photographers from attempting to determine whether a shutter is properly operated at, say, 1/200 seconds by comparing the flash duration produced by directing the camera towards the source of light while the shutter is tripped against, say, 1/100th of a second. Day and night vision. As stated previously in the duplicity theory, it is assumed that the retina contains two almost independently As stated previously in the duplicity theory, it is assumed that the retina contains two almost independently functioning receptors, the rods and the cones. The rods are more sensitive to weak intensities than the cones, but the cones are responsible for color vision. It is likely to expect then that data determined for the eye would be influenced greatly by the level of intensity at which measurements are made, since in one case we have operating rod vision, scotopic or night vision, and in the other cone vision, photopic or day vision. That such distinct sets of data are obtainable is the chief factor in support of the double receptor concept. For example, it is well known that the eye does not have a constant sensitivity for all wavelengths through the visible spectrum. The eye of a normal observer is far more sensitive in the central part of the color spectrum, green-yellow, than it is at the red or blue extremes. However, the exact placement of the maximum of sensitivity is a function of the light level at which the test is conducted. In figure 4 are shown two visibility curves giving the general pattern of the eye response to various wavelengths from 4,000 to 7,000 angstrom units. Curve S being drawn for scotopic visual levels, fainter than 10 to the power of 4 candles per square foot. In figure 4 are shown two visibility curves giving the general pattern of the eye response to various wavelengths from 4,000 to 7,000 angstrom units. Curve S being drawn for scotopic visual levels fainter than 10 to the 4th power candles per square foot. I can't tell if it's a minus sign or a underscore. Fainter than 10 to the negative 4 candles per square foot, and curve P being drawn for photopic visual levels brighter than 10 to the negative 1 candles per square foot. It will be noticed that there is a decided shift in the wavelength. It will be noted that there is a decided shift in the wavelength sensitivity when the illumination levels are changed. At high intensities, the eye is more yellow sensitive than at low intensities, where the sensitivity maximum lies in the green. This shift in sensitivity is the well-known Purkinje phenomenon, and it is the peculiar action of the eye which makes the almost non-perceptible light from a dark green safe light quite effective for film inspection once the eye has become dark adapted, and hence the scotopic vision is in full action. Another measurable difference in retinal sensitivity for scotopic and photopic vision is the change in sensitivity across the retina itself. Figure 5 shows a curve representing Figure 5 shows a curve representing the variation in Figure 5 shows a curve representing the variation in sensitivity across the retina for photopic vision. The foveal region shows a maximum sensitivity with a smooth decrease towards the periphery, the blind spot, corresponds to the point of entry of the optic nerve in the retina, where there are naturally no receptor elements. In figure 6, a similar curve is shown, but this time for the scotopic eye. It will be seen that the fovea shows the lowest sensitivity under low levels of illumination, and this is supposed to be due to the absence of rods in this region of the retina. It is interesting to observe that the curve in figure 5 follows rather closely the cone distribution across the retina. Adaptation. We have had occasion to use the term adaptation before defining it. Adaptation. We have had occasion to use the term adaptation before defining it. It will not be necessary for our purposes to present the mass action principles involved in the adaptation process, but suffice it to state that the eye becomes adapted to various light intensity levels after certain periods of time, chiefly because of an equilibrium that is established between the formation and destruction of the retinal visual purple, and because of the involuntary changes in the opening of the iris. The latter factor can account for a small amount of the total adaptation only, although in photographic literature one is often led to believe that the iris is the only factor in operation. When one speaks of the fully dark adapted eye, reference is made to scotopic or rod. When one speaks of the fully dark adapted eye, reference is made to scotopic or rod vision, while the expression daylight adapted eye refers to the photopic or cone vision. Visual acuity. The ability of the eye to distinguish fine detail is chiefly dependent upon the perfectness of the lens system and upon the limits of resolution imposed by the structure of the recipient elements of the retina. Actually, the seat of distinct vision is limited to a very narrow angle, 1 degree, corresponding to the coverage of the fovea. As we have seen, the cones in the fovea are closely packed, and each cone has a direct nerve fiber leading to the optic nerve. By reflexive action, the eye focuses its image upon the fovea, not upon the optical axis, as might be expected. Outside the bounds of the foveal image, the sharpness of vision is greatly reduced, as can be seen from the diagram in figure 7. The measurement of visual acuity is no easy job. The test procedure has to be carefully planned and many factors properly considered in evaluating final results. Visual acuity varies widely in different individuals, and results obtained with any given individual are greatly dependent upon adaptation. Type of test object, glare, nature of surroundings of test object, etc. But one outstanding fact is that the visual acuity and the ability to distinguish variations in intensities are closely related. A black object on a white surface is readily distinguishable, but as the background is made more and more gray, the discrimination becomes increasingly difficult. In testing for the minimum perceptible contrast, large areas are employed, one of which is varied in intensity so that the variable. In testing for the minimum perceptible contrast, large areas are employed, one of which is varied intensity so that the barely perceptible brilliance difference is established for different adaptation levels. Typical results are shown in figure 8, where Typical results are shown in figure 8, where SC contrast sensitivity equals LA over LB minus LO, in which LA is the adaptation luminosity, LB is the luminosity of the outside ring of the test object, and LO is the luminosity of the center disk of the test object. From these data, it is evident that contrast discrimination increases with luminosity. Thus, we can expect that visual acuity will vary with adaptation level and have a maximum at high values. The work of Lithgow, as well as others, shows this to be true. In figure 9, a curve gives the increase of acuity with luminosity. For this work, Lithgow used the international test object shown in figure 10, which is a broken circle having an opening equal to 1/5 the outside diameter of the circle. By orienting the test object at different angles and asking an observer to judge the position of the opening, a measure of acuity is obtainable.
But while reproduction from a pictorial standpoint seems best when it embodies the visual defects, modern practice is to try to produce lenses without optical aberrations. This is, of course, desirable from a scientific standpoint. However, so long as the trend exists, it seems that all attempts to relate the results obtained with such lenses to visual likenesses should be dropped.